Today is April the 4th, 2016. My name is Jane Reckelhoff, and I'm in the Senior Physiologist Lounge in the San Diego Convention Center during Experimental Biology 2016. Today I have the distinct pleasure and honor of interviewing Dr. Judith Bond for the American Physiological Society's Living History Project. Dr. Bond has been a member of the APS since 1978. She is the Evan Pugh Professor Emeritus at Pennsylvania State University in Hershey. Dr. Bond's research on protein degradation, and particularly her studies on a unique and complex metalloprotease system called meprins, was funded continuously by NIH for 35 years. Dr. Bond was also my graduate advisor, so it's a special pleasure for me to interview her today. So Judy, thank you very much for agreeing to be part of the Living History Project, and welcome. And if you're ready, I'd like to ask you a few questions about your career.
Absolutely.
So, according to your CV, you were born in New York City. And so tell us a little bit about your childhood and how you became interested in science.
Okay. Well, my childhood was a happy, good childhood in the public schools of actually Brooklyn, New York. I had a mom and a dad who were not academics at all, but my mom was interested in science, particularly biology, and she was sort of a frustrated biologist. When she was in college, it was during the Depression, she had to go out and get a job and then sort of do her college career at night. And she infused both of my sister and myself an interest in sciences. My dad was an insurance broker and never did graduate from college, but both of them were very interested in education. And education was something that they saw as integrating into America, they were second generation, so I was third generation. And it was the way you advanced and found a satisfying career. So my dad's contribution, though, was teaching me about competitive sports, particularly tennis at that time. But that I count as important too, because you know, you learn to win and lose, which is very important in academia.
That's right.
You learn to set goals and keep at it, get knocked down and come up again.
Yes.
So those two influences, I think, were very important, both the high interest in education and then the attitudes towards things. The public schools were very good in Brooklyn. And it was the Sputnik era. So we were racing against Russia to get to the moon, and science was encouraged. I had some very good teachers in the early days. So all of those things I count as very important.
Okay, all right. So you received your BA from Bennington, and then you pursued your master's degree and your PhD at Rutgers. So what was it about Bennington when you were there that convinced you to pursue an advanced degree?
Right. Bennington is a college that's based on a Dewey philosophy, which is learn by doing. And science is perfect for that, because that's the way you learn. You get into the lab and you do experiments and you get data and do things in order to learn and advance things. So even though Bennington was known mainly as an arts school, they had very good individual type programs in science. And so I attribute that as very important, again, to the science emphasis and also the love of the arts. I learned, I did take an art class and a dance class and went to theater, and that has lasted all through my life.
Liberal arts.
Yes, and the whole idea of a liberal arts education, I think, is very good. It was also an all-women's school, which for me was an important thing too. You didn't worry about how you looked when you went to class or what you said, if it would affect your social life or anything. So you just sort of could focus in and concentrate on your studies, and that was also, for me, a very important factor. I'm sorry to see women's colleges fading away, but at any rate, so all those things, Bennington gave me a very strong basic education. I went on to Rutgers, which was a much more technical school, known for its chemistry and its engineering, and but I never felt at a disadvantage because I had a very good basic level of understanding of chemistry and biology and physics and math. So was your question just for the BA?
Yes, just for the
Okay.
BA. And so, talking about Rutgers then, you worked with Dr. Bird, Dr.
Right.
John Bird there. And so tell us a little bit about your experience in graduate school and about Dr. Bird.
Right. Dr. Bird was a physiologist.
Yes.
Particularly interested in muscle physiology. And when I was in graduate, there was a good group of graduate students that went to Rutgers at the time I did, which is the early 60s. And Dr. Bird had some projects with muscular dystrophy going on, and I became interested in protein degradation in this disease, in muscle particularly. It was a time when there was a lot of advances in protein synthesis and very little known about protein degradation. But to me, that the synthesis and degradation work hand in hand to keep levels of things for homeostasis, important for all aspects of physiology. So we started working on that. It was a time when lysosomes had just been discovered really had a coming out with Judith, who won a Nobel Prize eventually for lysosomes. And so we looked into how lysosomes were involved in the dystrophic process. And it was a project in which we decided at the end of that it was actually the lysosomes were an after effect. They didn't initiate the problem of degradation, but they started degrading proteins as a consequence of other things happening. But it got me into the whole field of degradation, interest in proteases, which lasted a lifetime. And in graduate school, so I learned what discipline is about, what data collection is about, how communication was important, both orally and written, and so a lot of lessons learned there. However, by the end of my PhD time, I was thinking maybe I wasn't so good at research and I should go into teaching.
Really?
Yes.
That's amazing. I didn't know that.
So at that time, I got married and we moved to Vanderbilt.
Right, right.
And I went to look for a teaching job, but I found out I wasn't qualified for teaching. I didn't have any education courses, and I had no experience teaching. In graduate school, we didn't teach at the time. And transitioning into the PhD and master's degree, I met Janie Park, who was a physiologist at Vanderbilt, and she said, well, why don't you come and work with me? And I did. And then I fell in love with research and wasn't, as a consequence of the postdoctoral years, decided that research and teaching, going hand in hand, were where I wanted to go.
Well, good. Very good. What was the environment like at Vanderbilt then?
Vanderbilt was an exciting place. Earl Sutherland was there at the time, and he was involved in the discovery of cyclic AMP, which was quite controversial. People didn't understand how cyclic AMP could be a factor in so many different hormonal responses, because they had different tissues and different hormones had different effects, but they all sort of were mediated through cyclic AMP. But he had a very good group that was, we had a lot of visitors and a lot of interesting conversations and science. Now, I wasn't working in the cyclic AMP area, but that wing of the Department of Physiology at that time was a very strong and influential one. With Janie Park, I was working on a mechanism of action of glyceraldehyde phosphate dehydrogenase. So it was a pretty biochemical project. It was a very satisfying project, and we were dissecting what residues in the enzyme were important for kinetics and for binding, and we published a paper in the JBC and was very happy with the outcome of that. And that point too, I felt I was more, even though my PhD degree was in biochemistry and physiology, I more leaned toward the biochemical end.
Yeah.
It wasn't until later years where I started getting back to physiology.
Yes.
When you were
Yes.
associated
Yes,
with us.
that's right. That's right. So your first academic position then was at Medical
Medical
College of
College of
Virginia,
Virginia.
which is now Virginia Commonwealth University. I still can't get used to that. I always call it MCV.
Right, right. Well, many people call it Medical College of Virginia Virginia Commonwealth University,
yes.
so it hasn't gone away.
That's right. That's right. And so you started as an instructor and
Right.
rose through the ranks to full professor.
Correct.
And so what was your experience like? Because that was like 20 years that you were there?
Yes,
About,
I was at VCU for 20 years. Well, it's kind of interesting. I started as an instructor, and my husband started as an assistant professor, even though we both had the same credentials. So it was, I didn't realize it at the time, but there was a discriminatory,
Right,
a woman didn't need a job,
Yes, that's
was the
right.
philosophy.
That's right.
And
That's right.
so I started at a lower rank and salary, but, and also the other thing that I discovered was I was given assignments that were more, that the chair felt were more suited for women. So my teaching assignments were in the nursing school where most of the students were women. And I was given assignments in, charge of hospitality or something. There were a number of things like that. But, once you started working and getting into things, our colleagues didn't notice whether you were a man or a woman when they were discussing science.
Yes, that's
so
right.
I think and then they would invite me to participate in a course that was not necessarily only women's work. So I rose in the ranks there from instructor to professor over the years, and established my own lab, a lab of her own, and
which was very successful when I was there in the early 80s.
It's in the
Yes,
80s?
yes,
Yeah.
82
So-
to 85, yeah.
Okay, so I started there in like 68. And by the time you were there, right, we were up and running
That's
and
right.
had an exciting
That's right.
time.
A fun time.
Yes, we had discovered meprins.
Right,
And
right.
I had established some collaborations with overseas British scientists.
Right.
And a lot of exciting things happened.
Yes, I think you had just come back from a sabbatical with
Rob Bainin.
Yes, with Rob Bainin.
Yeah. That was,
No, it's
actually the
Strange Ways. You were at
sabbatical
Strange
was
Ways,
with Alan
yeah.
Barrett
With
at
Alan
Strange
Barrett,
Ways.
yes.
Yes, and that was also very important for career growth, sort of opened up my eyes to international science and ways to do. It's interesting, the British, we were in an institute called Strange Ways Research Laboratory, and it was very British. And so you didn't come in before 9 o'clock, and then you had tea at 10, or coffee or something. And then you worked for an hour or two, and then you had lunch. And often lunch was substantial, and then you went back.
Very civilized. Unlike
And then
we
3
do.
o'clock was tea again. And then everyone went home at 5. Now, for American scientists, this didn't work.
No.
So after the first few weeks, I was in the lab through most of the coffees and teas and lunches. But they got a lot done, even with a very different attitude towards work. And so that helped and was interesting and influential as well.
Yeah.
But I also met Rob Bainin, Dr. Bainin from Liverpool at that time, who was interested in protein turnover. And that turned into a very productive collaboration.
For a number of years.
For a number of years.
Yeah.
He came to the lab with some assay that he had for proteases, and we looked at it in diabetic mice, which I was studying at the time, and we found this high proteolytic activity in kidney, which we couldn't find anything about in the literature. And so we isolated it, purified it, started studying it, and that was the beginning of meprin biochemistry and physiology. And when you were there, of course,
we
had so many questions.
Yes, we had lots of questions. It was like the beginning, and so it was,
how could this happen? What is it? It was not a simple protease.
No, it wasn't. It wasn't. And we were trying to do the genetics of it and trying to see what chromosome it was on at the time, and so it was.
And we found some animals were deficient in the proteolytic activity. And you were involved in some of that.
Yes.
Found some of the meprin in the urine, which I remember you were involved with.
Yes.
And it turned out, you know, it was complex because there were it was an alpha and a beta subunit, two different gene products, and they were multi-subunit proteins, and they could form dimers and higher multimers, and they were membrane proteins, secreted proteins. So it was not your simple like trypsin protease.
No.
And that's why it was so interesting, and we had a hard time figuring it out, but it was engaging and had very good students such as yourself. So it was really fun.
Yeah. People at MCV that were important in your career, say mentors or people who were important.
Well, one was Galen Bradley, who is the chair of physiology, who turned out to be very encouraging and important in my career. He was about 10 years ahead of me, so he could, when it was time to go on sabbatical, he could let me choose where to go and encourage me to find the best possible group. Other people that were important were really colleagues who were in very different areas of research, but where we found overlap. And between, I think, Galen and a nurturing atmosphere, which also the department created, I think you could flourish in research.
We don't do sabbaticals very much anymore.
Well, Penn State has a policy, for instance, of every 7 years you can take one. And not a lot of people actually take them, which I think is a big mistake. Because it's hard, you know. First of all, if you have children, you have to arrange for all that, and a husband or a significant other. There are a lot of arrangements that have to go on. But I think it's a wonderful thing. This was another sort of life-changing experience for me, and fun. Besides living in another country and learning about another culture and forming relationships internationally.
So after MCV, then to Blacksburg, where you were the professor and head of biochemistry and nutrition.
Correct. It's the Department of Biochemistry and Nutrition. And that was my first administrative experience. And it was really a fascinating one. There, the strength of Virginia Tech is the undergraduate program. And it still is. When I was there were 350 undergraduate biochemistry majors.
Oh, wow.
I met Peter Kennelly, who was the chair of biochemistry after me. They have 750 now. It's a huge program.
That's amazing.
And it really, working with the undergraduates really is a uplifting type of experience, because they're so enthusiastic and they sort of bumble into something and get excited about things, and you just feel the future of science and biochemistry is going to be okay with all the problems that there are.
That's neat.
So I only stayed there for a short time, though, for a number of reasons. One, the administration changed and they decided to concentrate on their strength, which was undergraduate, but at the detriment of the graduate programs. And I thought the graduate programs was equally important and important for the undergraduates as well. And they were sort of squeezing out graduate education, and that and for research you need graduate students and postdocs as well.
That's right.
And then an opportunity came up at Penn State. Particularly, they were expanding at Penn State and Hershey. Jim Jefferson, who was the chair of physiology, and I had met at Vanderbilt when we were both postdocs. And he encouraged me to come on over, and I did. One thing led to another. And so my 20 years at MCV and 4 or 5 years at Virginia Tech then led to coming to Penn State.
So how long were you at Penn State?
20 years also.
20 years there too. Okay.
And also my work during a Virginia Tech time turned more medical. there were medical implications, and Virginia Tech didn't have a medical school at the time.
Right.
They do have one associated now.
Yes.
But I thought that would be a big advantage in looking at some of the functions of MEPRIN and its physiological implications.
So how were things then at Hershey and your career there? And
that was another very positive experience. It was a time of growth, so I was hiring 6 to 8 people. The department grew, and that was true of a number of basic science departments, and they invested in basic science. Hershey is in a very interesting location. You know, it's near Lancaster, Pennsylvania, and a lot of the patients actually are Amish related, and they don't depend so much on insurance. They pay right out, and they want to pay for services, and so it was on very good financial grounds. And it wasn't until some of the changes in reimbursement of medicine went on that they started getting squeezed and investing in the basic sciences, which was pretty late in my tenure there. But I enjoyed the atmosphere in Hershey. There's a very good collegial relationship between faculty members. Could attract very good students, graduate students, and so the program did well there, and the department. I also got interested in the MD PhD program, which I think is important to have people with a medical knowledge also know how to do research, because medical school itself doesn't give you research experience. And we had a number of successes through that program. I heard just yesterday when we were there, we had the MD PhD funded by the NIH, and then we lost that funding over some years. But yesterday they got refunded by NIH.
Excellent.
So there's now an MSTP program.
Very good. Excellent.
So that's good.
So did you have a specific focus of the faculty that you hired, or you hired just good faculty who were interested in a variety of things?
One of our, we had no specific area that we were looking for, and we were looking for the best scientists, but we wanted to get people who would integrate with other strengths and other colleagues at the medical center. So not people who were just so insular that they only had their own labs, because it was the time of thinking more in terms of team science or strengths of units rather than individuals. And that's a tricky way to go. But we hired people both in terms of proteomics and genomics, and all basic scientists, but that could relate to the clinical sciences or the new technologies. So we had really 2 different emphases. One was on protein chemistry, and one was on genetics. And that's the way.
So what do you think is your most significant contribution to science?
Well, the discovery of MEPRINs and its relation to functional aspects, I think, is at the core. The enzyme turns out to be important in a number of diseases and the immune response in general. And as you know, both in terms of kidney disease and intestinal disease, we have found very specific functions of the protease. And one of the functions in terms of the intestinal disease relates to inflammatory bowel disease. One of the subunits, MEPRIN alpha subunit, is a candidate gene for IBD.
Oh, really?
And particularly ulcerative colitis. And so that opens up a window that might have significance in the treatment of ulcerative colitis and
And the microbiome right now is getting much. There's lots and lots of research going on in the microbiome.
Absolutely,
so
and just how
perfect.
that is related, because pretty high meprin activity, particularly in the lower intestine, but small and lower. But so I think it's now being studied in other parts of the world, both in the USA. Moge Angera was one of these postdocs in the lab, and she has her own lab, and there's several posters here on meprin beta knockouts,
and good.
as well as in Europe, in Germany, and several other places.
Yeah, very good. So, service is also important, typically in a scientist's career, part of the important promotion and tenure package that you have to have. And while you haven't been very involved in service with APS, you were very involved with service with the American Society of Biochemistry and Molecular Biology.
Right.
You were past president of ASBMB.
Right.
You were also past president of FASB. And so just tell us a little bit about your experiences in service and just in general.
Oh, okay. Well, in terms of the societies, I mean, there are many aspects. There's journal service,
right.
And I was an associate editor of JBC for 14 years, which was significant.
That's a long time,
yes. But a very important one, because the quality of our journals, I think, is very important.
Yes, I agree.
But I've also reviewed papers for APS journals.
Of course, of course.
And so journal involvement. Grant reviewing is another thing that I've taken seriously, and actually I was not only on study sections, but I was in the NIDDK Advisory Council for 4 years.
Okay.
So that type of service. But one of the most enjoyable was the service as president of ASBMB and FASB. And there is, you're representing some of the best people in the world.
Right.
ASBMB is similar in size to APS, about 12,000 members. And I think the societies have a very important function. They are working for the scientists.
Yes.
They're working in terms of recognition of the importance of what we do, both to funding agencies and to Congress, which are important to our well-being, and which we've been very privileged to be supported by.
Right.
And they're working to help people in careers. As I know, you'll be a terrific president of APS.
Thank you very
You'll
much.
be doing all these
Thank
things.
you very much, yes.
And already have been. But the career development, the funding situations, and then there are lots of issues that affect us that, there are issues of, there are many groups in the country that are against using animals for research, for instance, and that's so critical to our professions that, you know, to explain why that is important, that you can't do everything on a computer. So I think issues like that societies take up and try to develop strategies to counteract some tendencies that could be detrimental to the, so a number of policies like that, and then training issues itself. There have been times when people have said there should be less graduate students.
Right,
well, some of us don't agree with that.
Yes.
And I think you need a group of people really thinking about it and making some important points of, first of all, research at a university is not the only career for a PhD. And unemployment is very low for PhDs, and what we need to fight for is the best possible use of the talents of these people, and societies take these types of issues on. And then there are things like meetings, important for people to get together and discuss their research and learn from each other. So I enjoyed that very much at ASBMB, and then the Federation is a federation of 30 societies now. So now it represents
You were a great spokesperson for all of us.
Now it represents 125,000 scientists and engineers.
Yes,
so
yes.
that's,
Yes.
and then when you go to congressmen and you say, I represent 125,000 people, they listen more
Yes.
than I have a lab and need to be funded.
Yes, that's right. That's right.
So, and
That's
so that was also a very satisfying experience. Yeah, so I think Janie Reckelhoff will have to think about it too. After me, one of my other students became president of FASEB, and that's Kenny Offerman.
Kenny Offerman, yes, I remember.
MD, PhD. Yeah.
Yes.
So we have now a tradition.
Yes, we do. We do. So we're at the experimental biology meeting. So what was the first meeting that you went to?
It was in the 60s, and it was in Atlantic City. I don't remember the specific year, but I think it was probably about 1965. And it was very exciting. It was very different in that people drove from all over the country. We drove in groups. We stayed in hotels in groups. It just opened your eyes to all the different science that goes on, and you know, it was a...
That was a FASEB, was that a FASEB
that
meeting
was
at that
FASEB
time?
meeting.
That was FASEB, yeah.
I don't remember how long, when FASEB transitioned into experimental biology, but it was not in the 60s or 70s, it was sometime after that. I think it might have been in the 90s.
I think so, yeah.
But it was, there was a time when FASEB meetings were like 22 to 25,000 people.
Yes, I remember those days. I do.
Do you remember that? And now, you know, now there are so many competing meetings.
Yes.
And small meetings, and people have to decide. You can't go to too many meetings. The big meetings have a very different value than the very small area-focused meetings. And here, you know, you can hear things that are outside your field by Nobel laureates and by and go to try to catch up on different areas that you're not familiar with. That's not always available with the
But I remember early on you organized those small conferences, like the proteolytics conferences that we had when you were at MCV.
Well, those were international.
Yes, they were.
So the International Proteolysis Society grew.
And those were very interesting. I remember as a graduate student going to those and being in awe of all these scientists from around the world that were there. It was
Science is an international enterprise.
It is.
And I think it's very healthy to be exposed to that. So did you go to the Williamsburg meeting, or?
I went to in Airlie. I went to the Airlie meeting.
So that was the first meeting I was involved with, right? That was about 1984.
Yes.
Yes, and that was wonderful. Proteolysis across the countries has come up to, you know, it's maintained a group of very dedicated people and grown in a number of countries.
So tell me, what advice would you give students today, those who are starting out?
Well,
especially in this time when, you know, funding is not so good and there's a lot of naysayers.
Right. Well, I think students have to have some faith that they will be able to do something with all this sweat and tears that you put into a graduate degree. But if they really get engaged in the work and the project and know they really like to do the science, I encourage them to go on. If they don't like it, it's better that they do something else. So I don't think science is for everybody.
No, I don't think so either.
But if they really sort of like doing it, I know I liked science. I didn't know what I was going to do with it. Then I think they have to keep their options open. They have to go to, you know, apply to a number of places and go to some place that they try to get into the best of the labs that they can find, or the most interesting, the most engaging, and then work very hard at it. I think that it is important to find your career path, and I encourage all students to get involved with this IDP,
yes, yes. Individual development plan.
There you go, individual development plan. That it is important that you start seeing what it is you like to do, what are your strengths, what are the opportunities, and to think about it earlier than we did when we were in graduate school, because there were many opportunities when we were getting out, and there are less so in the research university type of tenure track positions.
Right.
Although I don't think that they're disappearing, these opportunities that we've had, and there may be a little turn-up coming. I know at Hershey, when I left, the new chair hired 6 new people, and that was not in particularly good times, 2012.
Right. Yeah.
And I'm hearing more and more people are investing in the basic sciences. Francis Collins is giving a talk tomorrow morning about the importance of basic science.
Oh, very good. Very good.
To the society, to education, to science. And I think people are beginning to realize that, things like this CRISPR technology, that came from very basic science, and it has all kinds of implications for disease and medicine. So with the realization of how important it is, I think we're going to see more support from our congressmen and from funding agencies for very basic science leading to the clinical sciences. So I would say stick in there. If you really love teaching, you'll find a path. If you like communication, you can find a path.
Yes.
We need some senators and representatives
That's true.
who know something about science.
We need some support.
Yeah.
Very good, very good. I remember when I was a graduate student with you, you told me that there are 2 kinds of scientists. There are those who are brilliant, and there are those who are persistent. And you said you must be persistent. And so I tell my students that, and I say, and I'm not brilliant, but I am persistent. And Dr. Bond taught me that.
Well, in my experience, it's not necessarily the most brilliant students who are the most successful in producing science and in advancing science. So, yeah, that's pretty good advice.
Very good, very good. Well, thank you very much, Judy.
You're very welcome.
Please enjoy the rest of the meeting.
Absolutely.
Okay.